GenSight is a publicly traded company based in France, devoted to gene therapy and retinal disease, as well as CNS disease. With proprietary technology of mitochondrial targeting sequence that actively drives a gene into the mitochondria of the retinal ganglion cell. We have a seasoned management team, and by that I mean old. But the real value of a company is not its management, it is its assets. So let me tell you about what we do. We have 3 active phase 3 trials for Leber's hereditary optic neuropathy. And let me remind the group that Leber's hereditary optic neuropathy is not Leber's congenital amaurosis. Ours is an optic neuropathy, the most common mitochondrial disease, interestingly enough, genetically determined and driven, affecting young adults in the prime of their life, leading to irreversible blindness, with several genotypic possibilities, but one phenotypic expression. This is what the patient with Leber's hereditary optic neuropathy looks like: a peculiar, unique pathognomonic fundus photograph of peculiar surface telangiectatic vessels that do not leak on fluorescein angiography. We've had 2 clinical trials fully enrolled, rescue and reverse. Both are identical in terms of protocol. The reverse trial, which we've read out top line, allowed patients whose lost vision was between 6 and 12 months. The rescue trial, to be read out end of this year, is for patients who lost vision between 0 and 6 months. Let me share with you the results of reverse and see if I can place them in context. We had 37 subjects. There was 1 intravitreal injection. We compared the drug-treated eye to the sham-treated eye. And in fact, the drug-treated eye gained a logMAR improvement of 0.218, perhaps 11 letters. The atypical and unexplained observation is that the sham eye also improved to the same level of change. Very much counter to what we know of the natural history of this disease. What else did we see in the top line data? The temporal aspect of the retinal nerve fiber layer significantly was preserved in the drug-treated eyes compared to the sham-treated eyes. Essentially, the drug-treated eyes did not lose thickness of the retinal ganglion cell, of the nerve fiber layer. Remember, these are the axons that make up the optic nerve. This is the anatomy one would choose to protect if one wanted to intervene for this disease. And the protection of the temporal aspect of the nerve fiber was statistically significantly different. In addition, the ganglion cell volume of the drug-treated eye was also preserved, statistically significantly so. So the objective measures, the objective metrics in this trial showed protection of the neuroanatomy. And if replicated, arguably this could be the first demonstration of neuroprotection of the central nervous system axons in gene therapy in man. What else did we see? We also looked at contrast sensitivity with Paley-Robeson charts. And what we found was in this study, the contrast sensitivity of the drug-treated eyes almost doubled. Again, statistically significantly different. And for many of the neuroscientists, for many of the clinicians, for many subjects, contrast sensitivity is a visual function that is recognized and perhaps more relevant to the real-world conditions, as opposed to high contrast Snellen acuity measured in an eye exam lane. What about some other observations from this reverse study? If we look at the patients who came into the study on chart with better vision, and we compared best treated eyes to best sham treated eyes, we also found a difference in the visual acuity of that subgroup. 12-letter gain versus 4-letter gain, a delta of 8 letters for the drug group. On the other hand, trials are viewed as binary events, certainly by the market. This may be a simplistic analysis of a complicated trial. And when you don't meet your primary efficacy endpoint, your first question ought to be, did we choose the right primary efficacy endpoint? Is high contrast acuity the right metric for patients with Leber's hereditary optic neuropathy? In fact, if we had chosen as our endpoint the proportion of patients who wound up with acuity of 2200 or better, in fact, the GS10 treated eyes showed significantly more ability to maintain and achieve that level, with a probability of P of 0.0005. Had that been our endpoint, we would have had a successful trial. And just to put this in context, 2200 is where, and worse, is where these patients wind up on their own. So is our cup half full? Is it half empty? You get to decide. The primary efficacy endpoint was not met because both groups improved. We did show protection of objective measures of ganglion cell volume and temporal retinal nerve fiber. We showed a doubling of contrast sensitivity, a much more relevant endpoint for such patients. And for better patients who came into the study, there was an 8-letter difference between drug and sham. We have a second program, Optogenetics, which is the transfer of a gene encoding a light-sensitive protein into retinal ganglion cells to restore function when the photoreceptors are dead. We do this in a population of retinitis pigmentosa, but I say that our population chosen for this program is a stalking horse for AMD. We couple this with a device, a pair of goggles that enhances the transmission of light to the retina. Remember, this is loss of photoreceptors, preservation of ganglion cells. The axons of these ganglion cells are the optic nerve. When they discharge, a signal is sent centrally to central visual pathways. And we've got a phase 1/2 beginning, first in man, first subject to be injected in more fields at the end of this summer. So we have a company that's got 2 separate programs, has 3 fully enrolled ongoing phase 3 trials. Our third phase 3 for Leber's is a bilateral trial. We have an optogenetics program that will address a totally different strategy, taking all genotypes of RP, transforming the ganglion cells, bypassing dead photoreceptors into new photoresponsive cells. Thank you for your attention.